Here, we will dive in details of causes of hemolytic anemia. On broad classification, there are two types. First one is congenital, which means some disease or physical disability is present from birth. And second one is acquired. Here, the culprit lies outside the red blood cells. Congenital causes include hereditary spherocytosis, G6PD deficiency, sickle cell anemia, and thalassemia, while acquired causes include cardiac hemolysis and autoimmune hemolytic anemia. Let's begin with hereditary spherocytosis. It is an autosomal dominant inherited disease, which means that a person with a single copy of the mutated gene can develop the disorder. Hereditary spherocytosis, or HS, basically presents abnormality in membrane of RBCs. We can say it is built-in cell membrane defect from childhood. This is the normal RBC. In case of genetic defect, biconcave shape of RBC is lost due to decreased stability and decreased surface to volume ratio, resulting in spherical shape. Hence the name spherocytes is given. Sphero means spherical and cytes means cells. Let's see the details. RBCs possess plasma membrane protein. One of them is spectrin. Another protein which anchors spectrin to another cell membrane protein is called ankyrin. Transmembrane proteins can be of many types, such as band 3, protein 4.1, glycoproteins, etc. Among all, ankyrin and spectrin play a major role. If they misbehave and don't function properly due to some mutation, or if there is some kind of inadequate production of these proteins, cell membrane will lose its flexibility and won't be able to hold the biconcave shape. The altered shape of RBCs makes them hard to pass through the finest blood vessels, and they get stuck in sinusoidal capillaries of liver, spleen, and bone marrow, which increases their susceptibility to destruction by the macrophages, causing hemolysis, or premature destruction of red blood cells. Another possibility is splenomegaly, that is, enlarged size of spleen, which is due to entrapped deformed RBCs in the spleen. For diagnostic purposes, we will count on blood test to evaluate levels of RBCs, reticulocytes, and bilirubin. A peripheral blood smear will show characteristic small and spherical, means microcytic and darkly stained, indicating hyperchromic specks. Another tool is osmotic fragility test. It measures tolerance level of RBCs against changes in osmotic pressure. In case of HS or hereditary spherocytosis, when RBCs are exposed to low salt concentrations, they are more prone to hemolysis, which indicates increased fragility of RBCs. Eosin 5-millimite or EMA binding test. Here, EMA is basically a fluorescent dye, which when binds to protein band 3 on the membrane of RBC, produce fluorescence. In HS, reduced surface area of RBCs results in enhanced binding of EMA, which is determined by the fluorescence intensity of labeled RBCs. Additionally, acidified glycerol lysis test can also be performed, in which RBCs are exposed to acidified glycerol and rate of RBC lysis is determined. Normally, RBCs are less vulnerable to acid-induced lysis, while in HS, RBCs show increased sensitivity and a higher lysis rate. Last but not the least is genetic testing, where mutations linked to HS are identified. Now, let's jump in to see the treatment modalities for hereditary spherocytosis, which include folic acid supplementation for RBC maturation. If anemia is severe, splenectomy is performed, which is defined as surgical removal of the spleen to reduce RBC destruction. Well, some options come with the risks. Here, splenectomy increases the risk of certain infections by encapsulated bacteria, so immunization against pneumococcus and Haemophilus influenza type B and meningococcal vaccine should occur beforehand. Now we will be discussing another congenital cause of hemolytic anemia, which is G6PD deficiency. It stands for glucose 6-phosphate dehydrogenase enzyme. This enzyme protects RBCs from oxidative stress. In some people, there is an inherited genetic condition when this enzyme is inadequate, causing G6PD deficiency. The gene for G6PD is X-linked, so G6PD deficiency is much more common in males. Let's revise some previous concepts. Aerobic respiration includes 3 steps that are glycolysis, Krebs cycle, and electron transport chain. Glycolysis takes place in cytoplasm, whereas other 2 occur in the matrix of mitochondria. There is no aerobic respiration in RBCs. But why? Because they lose their mitochondria during development. So they undergo glycolysis and adopt pentose phosphate pathway that branches from glucose 6-phosphate and produce NADPH for their survival. Now let's dive in the details of mechanism of G6PD. G6PD is an enzyme that is responsible for conversion of glucose into 6-phosphogluconolactone and then into ribose 5-phosphate. In both these steps, NADP positive is converted into NADPH. Let's understand about the duties of these NADPH molecules. They basically help glutathione in fighting with free radicals and reactive oxygen species. They both are basically unstable and highly reactive species that react with other cellular components, causing damage to DNA and many proteins. The difference between free radical and reactive oxygen species or ROS is that free radical may or may not have oxygen, while reactive oxygen species must have oxygen atom such as H2O2 or hydrogen peroxide, HOCl or hypochlorous acid, and hydroxyl radical. Additionally, ROS is a subgroup of free radicals. Now we're gonna see how G6PD, NADPH, free radicals, and glutathione are interconnected. Let's begin with the glutathione. It has sulfahydryl or SH group in its reduced form. It donates hydrogen of SH group to detoxify reactive oxygen species, and as a result, disulfide link forms, inducing oxidized state. This oxidized state must be converted back into the reduced state of glutathione by gaining more hydrogen from NADPH, so they can protect cells from free radicals and reactive oxygen species. NADPH used here came from breakdown of glucose in presence of G6PD enzyme. If this enzyme is insufficient, there will be less production of NADPH and ultimately less reduced form of glutathione to wipe out reactive oxygen species. Buildup of free radicals and reactive oxygen species then damages hemoglobin, which will precipitate out inside cells and start binding to cell membrane, forming Heinz bodies. This makes situation more critical and renders RBC less flexible, and they won't be able to squeeze through capillaries, causing hemolysis and hemolytic anemia. Many people are asymptomatic until an acute hemolytic episode occurs. The triggers of acute episode can be some infection, few drugs such as sulfonamides, primaquine, and ingestion of fava beans. An acute hemolytic attack is usually self-limiting and only supportive care is required. A chronic hemolytic anemia is rare. Diagnosis of G6PD has following steps. First is medical history. It is to check for potential triggers as well as physical examination to look for signs of jaundice, anemia, or splenomegaly. Another common tool is G6PD enzyme activity test, which measures activity level of G6PD enzyme in RBCs via spectrophotometry or a fluorescence-based assay on a blood sample. Obtained result is then compared to reference value. G6PD deficient patients have activity level less than 20% of normal. Characteristic feature of G6PD deficiency is determined on blood smear by presence of Heinz bodies. Finally, there is genetic testing, where mutations linked to G6PD genes are identified. Now, coming towards treatment strategies. There is no specific cure for G6PD deficiency, but management primarily involves avoiding triggers that can lead to hemolysis. In severe cases, hemolysis can be treated by transfusion. Now, we will hover over another disease, which is sickle cell anemia. It is a genetic disease of RBCs where biconcave disc-like shape is altered into crescent or a sickle, which renders them rigid and hence undergo lysis, causing anemia. Hemoglobin type A has 2 alpha globins and 2 beta globins. Beta globin change their shape due to mutation in HBB gene. It is basically a missense mutation in 6th amino acid of beta-globin chain, altering a valine amino acid instead of glutamic acid. Now coming towards thalassemia, which is among inherited blood disorders characterized by abnormal hemoglobin production due to genetic mutation. Thalassemia is mainly of two types: alpha thalassemia and beta thalassemia, depending on which part of hemoglobin molecule is affected. As we know, hemoglobin has alpha and beta chains. If alpha chain is missing or compromised, it results in alpha thalassemia. And if beta chain is missing or compromised, it results in beta thalassemia. This disease can show symptoms from anemia to advanced complications such as organ enlargement and bone deformity. For treatment purposes, blood transfusion, folic acid supplement, and iron chelation therapy is required because iron accumulates in body on frequent blood transfusions. Let's see how iron chelation therapy works. When iron chelating drugs are given, they bind to iron present in blood and let the body excrete the bound particles via urine or feces. In the next part, we will discuss acquired causes of hemolytic anemia.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.